Hello, everyone. Welcome to today's webinar. My name is Christy Sullivan, and I'm the Secretary of the American Society for Cellular and Computational Toxicology. Today's webinar, like all of the webinars in our series, is presented by the ASCCT and the European Society for Toxicology in Vitro. And both of our organizations host regular meetings and pursue other activities to promote the computational and in vitro toxicology fields. If you want to learn a little bit more about either of these societies, I'd encourage you to go visit our respective websites, and we'd really appreciate your membership and support and involvement. As you're listening to the webinar, you are able to add questions. What you do is go down to the bottom of your screen and click the Q&A box, and please put your questions for the presenter while they're speaking in the Q&A box. If you need to reach us for a technical issue, you can send us a chat message, but the Q&As, please put them in the Q&A module. And then each presenter will have their slides and their lecture, and then we'll have a Q&A after each presentation. Your lines are muted, and so we'll facilitate all the involvement through the Q&A module. We are recording this webinar, so the recording will be posted on the AICCT website a few days from now. And if we're able to make the slides available, they will be there as well. I just wanted to put a quick plug for the AICCT's annual meeting this fall. It's going to be October 12th through the 14th. It is going to be a virtual meeting, so anyone can participate. We have some really nice themes around the overall theme of the practical applications of new tools in toxicology, including AI, omics, new approaches to validation, solving 21st century challenges, and we have some really nice lineup of speakers to talk around those themes. You can submit abstracts in order to apply for an oral talk or a flash poster presentation by July 23rd, so that's this Friday. If you want to submit an abstract for a poster presentation, there will be a virtual sort of standard PDF poster session. You can wait a little longer, and that deadline will be in September. In August. So please visit our website and find out more about that, about the meeting. Also, I wanted to mention that the ESTIV organization is having its next meeting in 2022, and they're going to be opening abstract submission in December of this year. They also have 2 awards available for a Best Poster Award at the World Congress on Alternatives in Animals and Science. That is a virtual meeting later in August, but they have an award for the presenters there at that conference, and then an award for Eurotox as well. And you can find out more on the ESTIV website right there. So I'm going to go ahead and introduce our presenters today. First, we're going to have a talk on the links between air pollution and COVID-19 in England from Marco Travaglio from the University of Cambridge. Marco is currently pursuing a PhD in toxicology at the Medical Research Council Toxicology Unit at the University of Cambridge. Previously, he completed a Master of Science degree at the University of Nottingham and spent 1 year in industry working in the Translational Neuroscience Unit of Eli Lilly. His research in Cambridge is part of a collaboration with AstraZeneca and aims to deliver novel mechanistic insights into the vulnerability of certain types of neurons in neurodegenerative diseases. His project involves the use of both embryonic and induced pluripotent stem cell-based model systems to study the onset of diseases and their subsequent pathological manifestations. Following the COVID-19 outbreak, Marco and his group devoted their research efforts to uncovering novel risk factors for COVID-19 illness and death using a combination of in silico tools. The second presenter is Maria João Amorim. She's going to be talking about COVID-19 through adverse outcome pathways, building networks to understand the disease. Dr. Amorim is a virologist and group leader of the cell biology of the viral infection laboratory at the Instituto Gulbenkian de Ciência in Portugal. She started her research group in 2012 following a PhD and 3 postdoctoral research projects at the University of Cambridge and the National Institute of Medical Research in the UK. The main interest of her laboratory is to understand viral host interactions, identifying host factors and pathways essential for viral replication, and virulence factors that impact viral disease. Using influenza as a model, they focus on how liquid organelles and liquid-liquid phase separation influence the viral life cycle. Since SARS-CoV-2 pandemic started, she's been working on novel methods of identifying actively infected people with SARS-CoV-2 and understanding how the viral SARS-CoV-2 evolution shapes the ability of the host immune system to avoid reinfection. Most recently, she's been using adverse outcome pathways to understand COVID-19 through participation in the Chow project, which she will tell you about in our second lecture. So with that, I will go ahead and give the floor to Marco.
Thank you. Thank you so much for the presentation. I'm going to share screen now. Can everyone see my screen?
Yes, looks ready to go.
Perfect. Okay. Right. So just as Christy said, my name is Marco. I'm a PhD student at the University of Cambridge. I work in the MRC Toxicology unit, which is a department within the School of Biological Sciences. My supervisor is Miguel Martins, and today I will be presenting our data in relation to COVID-19 and air pollution in England. So the first slide is just a way for me to set the stage and provide some background on the study. As you can see, the analysis on air pollution and COVID, which is the purpose of our study, started really in March 2020, perhaps April. And this is when a paper was published. In fact, it was at the beginning of April, looking at correlation between air pollution data and COVID-19 cases and deaths in Italy. This paper was published around the first week of April. We looked at that and we're trying to complement it with data sets coming from the UK. The reason for that is because during the lockdown, a lot of the laboratory and institutes were shut in the UK, and we're trying to provide some help to the COVID-19 scientific community by engaging in some computational work. So that all started around spring, and then we trying to not only analyze data sets coming from the UK, but also refine our models over time, particularly during the peer review sort of refinement period over the summer. And we published our study eventually in October 2020. In terms of the overall aims of the study looked at the relationship between air pollution and COVID-19 infectivity and mortality in England. I will explain why we decided to look specifically at this. And one of the things that I should mention at this point is that although the study started as a sort of ecological investigation, so we looked at population levels, cases from COVID-19 data, and also deaths from COVID-19. We also utilized this UK Biobank cohort that provided more granular data on the spread of COVID-19 and also on other comorbidities and measures that we were able to analyze to sort of reinforce the association with air pollution. In terms of the background, I mentioned at the beginning that there was this paper that was published at the beginning of the pandemic, really, in Europe, that looked at the correlation between COVID-19 and air pollution. I think this paper was very topical and very important for us because it was the first paper to actually show or suggest that there might be a correlation between these two factors. And the other reason why we looked at it was because I'm sure you can remember that the epicenter of COVID-19 spread in Europe was in fact northern Italy. So there was a lot of attention being directed to that specific region at the beginning of the outbreak. And so we wanted to understand a little bit more about the dynamics of the spread of the virus and also whether there could be any factor that could facilitate the diffusion and the spread across other countries. So we looked at air pollution data, and in fact, I'm going to show you some of the resources that we engage with to be able to pull up some air pollution data. Me and other students in the lab, we looked at publicly available sources, and this is a very important strength of our study. We essentially scrape the web and try to understand where the air pollution data could be found, as well as COVID-19 data. We steered away from sort of, well, what we tried to do was to look at official government data sources, particularly for COVID-19. You might remember that the testing rate in the UK was high enough for us to construct some robust models of this correlation between air pollution and COVID-19, but at the same time, a lot of asymptomatic cases were not being tested. And we started to realize that that could be an issue early on during our study, because we wanted to reflect this correlation between air pollution and COVID. And so one of the resources that I mentioned earlier was a UK biobank that you see here indicated at the bottom in the module, the blue module in the middle. And this was really a strength of our study, because in the biobank, as I will be describing later on, all the individuals considered to be positive, all the individuals that adhered to the COVID-19 testing module were tested for COVID-19, regardless of whether they were symptomatic or asymptomatic. So we had a little bit more sort of confidence in our data, looking at individual level spread of COVID-19. In terms of the first part of the study, I think it's safe to say that when we submitted our preprint, we only looked at regional levels of air pollution. So this is a very high level sort of analysis that we did. We didn't go into too much depth. We looked at different regions of England, and we saw this striking correlation between the number of cases that you see in blue in the top left map of England, and then deaths, which are reported in red in this heat map. And as you can see quite clearly, the highest number of cases and deaths was reported in London and the Midlands, as well as the north part of the country. And these were the areas that also were found to be characterized by very high level of nitrogen dioxide and nitrogen oxide. These are two of the most popular gases, toxic air pollution molecules that are found in the sky. And in fact, these are were found to be of very high levels in those regions. So when we looked at this data, we started to understand that there might be a correlation, but we also wanted to test this hypothesis with more granular data. And that's the reason why we then went on to refine our model. So what we did next is part of the sort of refinement of our initial analysis. We understood very early that looking at those 7 data points wasn't enough for us to draw any conclusion. And so we tried to improve our model, this time looking at what happens when we increase those data points from 7, which is the number of regions that I showed in the previous slide, to about 304 data points. And that's the image that you see in the top right. The reason why it's 300 is because that's approximately the number of local authorities that you have in England. I shouldn't say the UK, because our study focused on England. And the reason why we now looked at that was first because we were able to increase the amount of data that was available. And then secondly, because looking at the local authority, we were able to match the air pollution levels to other confounding variables in addition to COVID-19 cases and deaths. As you can imagine, a very important one when looking at air pollution levels is population density. So we were able to account for that in our refined model. So this time, when looking at the correlation between air pollution levels and COVID, we also looked at the density at local authority. We also looked at the mean age, as well as socioeconomic and socio-demographic factors within each local authority. And when we ran these models, this time looking at, again, more data points, but also more confounding variables and more air pollutant species, which are indicated on the right, we again found a strong correlation between air pollution levels and COVID-19 infectivity and mortality rate. So these 2 graphs that you see on the left, they show that the 2 air pollutants that were found to be most strongly associated with COVID-19 cases and deaths were nitrogen oxides and nitrogen dioxide. We did not find a strong correlation at this stage with this type of analysis with PM2.5, so that's particulate matter, and 2.5 and then PM10. And ozone always tends to have this sort of opposite relationship with nitrogen dioxide. So we were not too surprised to see that ozone is negatively associated with COVID-19 cases and deaths compared to nitrogen dioxide. In terms of the methods, I was providing a little bit of details in the previous slide, but just to be a little bit more accurate here, the cases that we were looking at this stage were about 100,000 cases. And these were all cases reported in the first phase of the pandemic. So we're looking at the sort of early spread of the virus in England. And then we looked at just over 30,000 deaths recorded in the first few months of the pandemic. And we matched that to air pollution levels reported up to and including 2018. Unfortunately, at the time of the analysis, data from the year before the pandemic, 2019, was not available. So we were not able to match air pollution levels to 2019. However, we did find historical data in the 5 years before. So we had two types of analyses, which are shown in this graph. One looks at air pollution level just in the year 2018. And the second one looks at historical data to understand whether potential fluctuations in air pollution levels could account for any differences between these single air and multi-air models. That's how we call them. And as you can see from the results, there was very little difference, meaning that perhaps those fluctuations in air pollution levels did not account for any major difference. The other thing that I should mention, and I said this earlier, is that we're looking at the first phase of the pandemic. So all the cases and deaths were reported up to and including April 26. In terms of what we did next, so this is our sub-regional analysis. But as I mentioned earlier, it's very important when looking at air pollution data to understand what happens at the individual level. In fact, after we published our preprint looking at this correlation between air pollution and COVID, there were many other studies that identified a similar relationship in other countries. But what was striking is that at the time, it was very difficult to understand what happens once you have patients and once you match comorbidities from individuals to COVID-19 infectivity and mortality in the context of air pollution. So that was a big question that we had, and we were lucky in that because we already had access to this biobank called the UK Biobank, which I mentioned earlier. And this biobank provides data for over 500,000 volunteers across the UK. You also have clinical data, so you have historical medical records with each individual. And at the time of the analysis, we had about 1,500 participants, 1,464 participants that were tested for COVID. Of these participants, 669 were positive. So we had a large enough sample to understand whether air pollution level could have any effect on these individuals that tested positive. So what we did next was to model the postcode that was reported on the biobank for each individual and understand more about the exposure to air pollution levels for those individuals that tested positive. The scientific question behind that was if we can understand whether those individuals live in areas with high air pollution levels, maybe that can give us some information about their vulnerability to COVID-19 infectivity or deaths. So in this model, we included about 16 confounding variables. So we had data that we reported as social demographics. So that includes age as well as other information, but also socioeconomic factors. And among those, we had quite granular data to the point where I think one of the variables was how many family members they have. So how many people they share the house with, or what type of house do they live in? Is he a mansionette? Is he a flat, or is he a two-story house? So we did have a lot of information on these individuals that we could then match to air pollution level. And the finding from our study, and this is in fact the biggest change that we saw, was that with every single unit increase in PM2.5 level, we found an increased risk of infectivity of about 12%. which is interesting because at the sub-regional level, we were not able to pick up this effect driven by PM 2.5. But then when we looked at individual level, we did saw this difference. So the conclusion, or at least what we discussed later on in the study, is that individual level data might be necessary to unmask the effect of air pollution when looking at COVID-19 infectivity. And in fact, this was a bit of a warning signal to governments that were about to lift restrictions to traffic and mobility later on in the summer. So we did have several conversations with different government officials. The main finding being that air pollution level can be associated to COVID-19 spread and mortality. Of course, this is a correlation, and I do want to stress this point because it's been asked a number of times after we published our study whether there is a sort of causal effect. And I think unless that is proven in a laboratory setting, I think it's hard for us to make speculation on that. But I think it's possible that air pollution level can exacerbate mortality from COVID. And it's also something that, at least for infectivity, has been proven in other studies after our publication. There are some reports showing that particulate matter particles can sort of transport the virus based on perturbation in the sky and other factors. So this thing can happen, and I think it's something that should be discussed in the scientific setting. In terms of the recommendations and sort of engagements with the government, we have talked to a number of MPs after the publication, and we were lucky enough to be in contact with the Cabinet Office as well in the UK to provide more guidance and sort of discuss our data in the context of the reopening of the economy as the cases starting to decrease over the months after we published our study. And one of the most rewarding outcome of our study was that in the wake of our publication and other studies that came after that, the New Delhi government introduced a campaign to reduce traffic. And that decision was made purely based on the science showing correlations between air pollution and COVID-19. And I think that is definitely one of the most satisfying sort of results of this study. At the same time, there were reports showing that starting from September last year, as the traffic started to build up again in the UK, the air pollution was roaring back. And that was just before we had that massive second wave and then a third wave in January of cases from COVID-19. So there is speculation that could have been driven by increased air pollution levels, but at this point, that remains speculation until it can be proven. So I just wanted to thank everyone for your attention. I think I'm more than happy to take any question. And then I just wanted to give some acknowledgement to some of the team members involved in the study. We've been extremely lucky to report to a number of newspapers to talk more in depth about this study. So if anyone has any question, I'm more than happy to take it. Thank you.
Wonderful. Thank you, Marco. That was a really interesting presentation. Everyone add your questions to the Q&A if you have a question for Marco. We have one already. I was just about to ask one, but we'll go with this one first. It says air pollution is usually linked to a large concentration of people. So I guess they're saying there's lots of high population in areas with high air pollution. And can we really say that PM 2.5 is responsible for increased infectivity? I guess they're saying maybe it's just that there are lots of people, in an urban area.
Absolutely, and that's in fact one of the points that was raised soon after we published the preprint. So if we go back to the original study, the original study mainly revolved around these results at the beginning when we looked at regional correlations between air pollution levels and COVID-19 cases and deaths. And of course, the question that was, can we really say that there is a correlation that without looking at how many people there are in specific regions of the UK or England? So the next phase of the study looked at a number of confounding variables that we introduced into the model, one of them being population density that you see here listed. So to answer your question, we did account for population density in our model. So even after ignoring the fact of population density, those results remain consistent.
Okay. What would be the effects or interactions between the virus and the internal metabolizing enzymes like cytochrome P450s?
Is that a question in relation to, I guess, air pollution?
Probably.
I don't think I can answer this question, to be honest.
Yeah, maybe whether whether the virus affects detoxification mechanisms is behind the question.
I think it's a very interesting area, so I welcome that question, but I don't think I have the knowledge to answer it. I think I will just speculate any answer, so I can't take that question. Okay, the general question. I see that reply.
Yeah.
It finds me unprepared on that.
Well, I have to note that after reading your bio, your area of expertise is different than this work here, and I'm just curious about what that experience was like. Kind of launching into a new area and looking into some new methods to do your science during the lockdown.
I think the short answer is that it was really exciting. I think everyone trying to refocus after the pandemic, and we all trying to do our best, pushing the boundaries of research. And I think I'm thankful to our supervisor for giving us the chance to do this. It did start as a sort of analysis exercise for us. We were just looking at any potential factor. In fact, we engaged in a different type of study after this one, and we published that one too, looking at another type of risk factor for COVID-19. I think the bottom line there is that when you are a PhD student or you're a scientist, you do have the skill to contribute to the fight against the virus, then I think everyone should do it.
Wonderful. Okay, so there's a question about the pollution measurements in 2018 with the COVID pandemic 2 years later. Do you know if well, I guess there's two questions here. So there's that, is that right? And then do you know if the virus bound to particulate matter is viable and infective? So those are two very different areas.
Yes, so to answer your question, Emanuela, I think this is a very good question. In terms of the air pollution levels from 2018, I think that is definitely one of the limitations of the study. And unfortunately, we didn't have any other means to look at air pollution because all the data that is recorded and stored in those databases is only available about 1.5, sometimes 2 years after, because of the processing of that raw output. So we didn't have access to it, but ironically, after we published the paper, then the data from 2019 became available. So we could have done it, and I think we did start some follow-up analysis, but we didn't pursue it all the way. What is reassuring though, and this is the reason why the paper was eventually accepted, is that we account for that anomaly by looking at historical data from air pollution. So based on the finding that the air pollution level didn't change dramatically over the 5-year period between 2014 and 2018, then we're quite confident that we're looking at the right thing with the 2018 air pollution data. In terms of the second question, there is evidence published from Italy and other countries that the virus can be bound to particulate matter. In terms of whether it's viable, to my knowledge, that remains an open question. So we do know that there are traces of RNA on the particulate matter of the virus. We know that it can be carried across, but we don't know whether it remains viable and people can actually be infected. My guess would be that, yes, that can happen, but it remains to be proven.
That's really interesting because it's, sort of maybe an additive effect where you have the particulate matter affects people's respiratory and immune systems and ability to fight the virus, and you have the virus on the particulate matter. Very interesting. I was wondering, you touched on this a little bit, about the mechanism of why people in areas with higher particulate matter might be experiencing increased infectivity. Can you say anything about that, or did you look into that at all?
Yeah, so that was an interesting finding from our study, and we're trying to understand a little bit more about it. And we do know that air pollution can cause those sort of inflammatory responses in the body. But what is more interesting is not the acute reaction of the body, it's more that sort of chronic response in those areas where air pollution levels are really high. So we did start looking into that, and in fact, we started sort of putting together all the findings to make sense of that. And there are reports out there showing that PM2.5 is critically involved with that. And in fact, one of the most authoritative studies that has been done in this field, looking at PM2.5 and COVID, came from the US, and it was published by a group at Harvard that looked specifically at PM2.5, and their findings were very similar to ours. So our percentage increase in vulnerability was 12% in our study. And in that study, they had an association that was reported to be 8%. So basically, any single unit increase in PM2.5 could lead to an 8% increase in deaths, if I'm not mistaken, in that study. So yeah, I think the key there, the link, is that inflammatory response that it causes in the airways, which can then exacerbate the infection of COVID in your lungs, your airways.
Yeah, it's certainly consistent with all the other modulating factors in terms of sensitivity or to the disease. One last question real quick, and then we'll move on to the next presenter. Just wondering about PM10 and whether you found anything relevant there.
So PM10 wasn't significant in our analysis, but I will guess that in terms of the toxicity, that would be quite similar to the PM2.5. We do need to be careful though, because there are some reviews out there that say that because of the size of those matters, so we're looking at under 2.5 microns for PM2.5 and over 2.5 microns for PM10. The size of those molecules or those matters can help them to propagate further into your airways. So the type of infection resulting from high level of exposure to PM2.5 might be different, because those particular matters can penetrate deeper into your airways and accumulate. So yes, there might be different responses depending on the individual species.
Great. And then thanks to Emanuela Corsini, there's some extra reading here. I've just popped that into the chat. So you can all see that reference or that suggestion. Thanks, Emanuela. Thank you, Marco Travaglio, for your presentation, for answering our questions. And we'll move on to Maria João Amorim.
Hello, I can't start my video somehow.
Maybe put up your slides and then we'll see what we can.
Okay, that's okay.
Oh,
good. There I am. Okay. So, hello everybody. Let's just put the slides up. So I'm here to present the work by a really big crowd of people, and it's the modeling of pathogenesis of COVID-19 using adverse outcome pathways. So what's the complexity of the problem, and why did we felt the need that this was important? Well, the complexity of the problem, we all know. We have a new viral infection that is very heterogeneous in the outcome of the disease, and it's very heterogeneous in the way that it affects different organs and that affects individual people. And so there's the necessity to try to understand it. And within this heterogeneity, it also follows a quite particular time frame in which there is from symptom onset until resolution of infection or more serious outcome. There's a time frame that is associated by acute infection until week 2, and then the complications could start with post-acute hyperinflammatory illness and later sequelae that even include long COVID. That is something that we don't understand. So this is a multi-layer, highly complex process. And so since the onset of SARS-CoV-2 pandemics, there has been an unprecedented level of literature that has been published even without peer reviewing, which is fantastic because it allows for scientific knowledge to be enhanced and advanced faster. But at the same time, there is the necessity to be able to understand exactly and extract the important information. So in order to do this, the Joint Research Center and Penny Nymark, they've decided to join forces in order to start using adverse AOPs in order to try to understand and put in practice the COVID-19 disease. So the idea here is that to go through the molecular data and up to the downstream consequences, in which there is the virus is considered a stressor that triggers a molecular initiating event, a particular cellular response, tissue and organ response, the response of the individuals, the response of the population. And so we are trying to look into these, as I said, several different layers of complexity. So in order to do this, the Shao crowd was created, and those are the stars of the entire project. And this is a multidisciplinary, cross-disciplinary panel of people that that is able to read and understand and work in some cases on the matter, and that through being divided in different working groups that I'm going to tell you a little bit further a long way, they produce knowledge, and this knowledge is either placed at the AOP wiki in the form of key events and particular AOPs and key event relationships, but as well the publishing teams that publish in peer-reviewed journals. And the crowd and their working groups generally meets every 2 weeks, every 3 weeks, and the entire crowd meets on workshops to decide the direction of the project. And there is a coordination group that oversees the direction of the crowd and that regulates and organizes the entire events. So basically at the moment there are over 65 scientists from 40 different organizations worldwide, and this is the acknowledgement slide already, which that involves a lot of people, a lot of different expertise, and that are important actually to understand the complexity of the disease. So basically the way it works is that there are the AOP developers, and these could have and display a different range of participations, from being full participants or belonging to the coordination group, for example, to being Shao junior collaborators or consultants. And there's of course the AOP users, which are scientists, clinicians, and policymakers, and so then there's the Shao crowd. And the Shao crowd, as I said, is overseen by the coordination group, and then it's divided into 6 other working groups, which is the literature review group, the hub lung AOPs group, other organs AOP group, the neuro group, the multi-scale impact group, and the modulating factor group. And so within what I'm going to tell you is about different progressions of all these groups, but in particular the group that I belong to, which is the lung and the hub AOP group. So basically in the lung and hub groups that have decided to join forces because they wanted to set the lung as the initial framework for everybody to be able to use not only the key events that are quite initial for the development of the disease, but also to provide some sort of basis for the development of the other AOPs. So basically here we divide them into the initial replication of the virus and the initial events that can lead to deregulation of the way that each organism is able to resolve the disease, to then what we call hub-related AOPs and key events that increase the inflammation, that increase that spread the virus and affect other organs as well. And then this leads to acute respiratory distress, to lung fibrosis, to coagulopathy and thrombosis and multi-organ failure. So how is it possible to capture all of these in AOPs? And that is the really tricky question to answer. So this involves a series of people, and there's the name of everybody that is involved here on the particular, on looking at particular aspects. And this is the crowd of the lung and the hub groups. So it's a really nice crowd to work with. So basically what we are aiming at the level of the lung is to try to explain how does the viral infection leads to difficulties in breathing, pneumonia, extensive alveolar damage, capillary congestion, necrosis of pneumocytes, interstitial and alveolar edema, type 2 pneumocyte hyperplasia, and excess surfactant ischemous metaplasia with atypia that could then lead to the adverse outcomes of acute respiratory distress, lung fibrosis, compression of the lung tissue, damaging pulmonary capillaries, and death by respiratory failure. And so in relation to the lung itself, we already have some AOPs on the wiki that are here described, and that I'm going to talk you through in some detail, just to highlight what we have been doing and really ask you for some feedback and also for your contribution. So one of these key events leads to lung fibrosis and is divided at macromolecular level with the binding of the spike glycoprotein to the ACE2 receptor as molecular initiating event. Then at the level of the cell and tissue, there's the epithelial cell increasing in angiotensin 2 and increased plasma angiotensin 2 levels and increased collagen accumulation leading to lung fibrosis. But what I would like to call your attention to is that this first, the molecular initiating event, it can be used by many different AOPs. And so the way that it is done, it's with a high detail on the key event describing it and on the key event relationships that then can link different AOPs. And we can see here just a demonstration of the species that can be infected by SARS-CoV-2 and the type of infectivity that is produced, and the literature and the references. And within that, it's also accompanied by some structural data of both spike and of ACE2 binding and the consequences of this effect for viral entry. And so it's no surprise that this molecular initiating event is actually being used by different AOPs and is actually being cited in many different AOPs that are here described. And so the idea here is that it is possible to actually understand the complexity of the disease, because the different AOPs that are built can actually be like Legos that we can actually use and apply in different AOPs. And these could also include, and very frequently include, key events already and key event relationships already on the wiki. So this also happens in another example that here employs the same molecular initiating event, but then here leads to viral entry in cells and viral gene expression. And so this is another example of the viral entry and the way that the virus replicates in the cell that can also be extensively used and extensively applied on other AOPs. In this case here, leading to lung, heart, and kidney disease, but mostly acute respiratory failure. There's the increased pro-inflammatory and pro-fibrotic mediators leading to inflammatory immune responses. So basically, within this particular AOP, I would like to refocus and highlight again the initial events. For example, the increased susceptibility to viral entry is described in a way that could be used and applied to many different AOPs, because it takes into account many different cell types, model cell types, as well as cells in the body, and it also takes into account the different host factors that mediate that can mediate and contribute to viral entry. So basically, SARS-CoV-2 spike protein, which is the protein at the surface of the virus here, that is able not only to mediate the viral entry, but also to allow for recognition of antibodies, can access the cells via two pathways. And I'm not going to go into detail for the sake of time, but just to tell you that both these key events, as well as the key event leading to viral replication, can and are being used in many different AOPs, as this example here, where the same molecular key event is used, the same viral entry mechanism is used, but here we started looking at the antiviral response that the cell can or cannot or is antagonized by the viral infection. Again, here this would have implications in viral replication and in the way that COVID-19 infection could be proliferated, which provides a loop. And so also for the interferon response, there's the same level of detail in which we have all the different orths of the virus, all the different proteins and their function within replicating, their function for replicating in the virus and their role in innate immune evasion. So this is quite a complex way to actually provide information and make it easier, because each small contribution can actually become part of a very big contribution overall. So two other AOPs within the lung, one leading to decreased lung function through alveolar collapse and through loss of capillary membrane integrity, and the same one leading to lung fibrosis through deposition of the MC. So a final one, just to go on to the same on acute respiratory distress syndrome, that is developing new key events on toll-like receptor and the way that they control systemic inflammatory responses. So then from here we go to the hub part, which is the part that can mediate a lot of the effects that are damaging to the whole body and that avoid resolving the inflammation and avoid resolving the viral infection. And as you can see, then this starts becoming more and more complex. So within the hub group, we look at what is already known and already placed at the wiki, for which we use many different key events. And we see how there's a process within the process of inflammatory activation and response. There's not only increased pro-inflammatory mediators and all the process leading to this, but also there's damage repair and return to homeostasis. And here within COVID-19, we know that what tilts or unbalances the body to worse adverse outcomes is deregulation of all these different steps. And so this is where we dig in and where we try to understand. So in one sense, we look at the activated bradykinin system leading to hyperinflammation, which is something that is extensively reported in literature, but we really analyze it. We really analyze how the experiments have, how the key events have been measured, and try to make sense of how it can contribute to the disease. And so we have here the bradykinin system leading to hyperinflammation. We then look into detail on thromboinflammation. And here the process, as you can see, is highly complex. But basically what we are focusing is the fact that there's platelet neutrophil interaction that is increased, and that this can lead to platelet aggregation and blood clotting initiation events. And so within the AOP, it starts and it tries to bridge coagulation and inflammatory related AOPs, focusing particularly on the platelet neutrophil interaction and how this can contribute to different outcomes of inflammation and coagulation. Okay, so and this is what we are doing at the level of the lung and the hub groups. But then there's another entirely different group, also formed by a different crowd, that and some overlapping crowd, that is the multi-organ group. And here this group is looking at the direct damage provoked by the virus, which is the direct effect, but also indirect pathways mediated by the hyperinflammation, by coagulation, by other effects that COVID-19 can have on other organs besides the organ. And within this second indirect effect, there's here this trying to look also at possible treatments and effects within the microbiome that could help and assist on resolving the hyperinflammation and the different affection of the different organs. So another group is the neuro-AOP group, and this group focuses only in the brain, looking into anosmia, encephalitis, stroke, and epilepsy. So also building a series of AOPs to be able to explain how these adverse outcomes can happen, and trying to link them to COVID-19 progression of the disease. Then there's the literature review group, from which Christy Sullivan is also one very active member, that are actually doing a pilot study to look and be able to retrieve literature that contains information about neurological output of COVID-19. And so they are using, and there's the full text screening is using this Swift active screener, in which the literature is retrieved. There's a curation and analysis, and some non-relevant papers are excluded, to then be able to, each full text is reviewed independently by at least 2 reviewers for inclusion or exclusion. And so there's a quite detailed analysis on how we can use the literature and how we can make use of the literature in order to feed onto the different groups. And this is very important as well. And another group that I am particularly fond of is as well the modulating factor groups. And these look at particular factors such as sex, age, comorbidities, associated comorbidities, lifestyle factors such as diet and smoking, environmental factors such as the ones that we just overheard in the previous talk, drugs, genetic factors, microbiota, all trying to understand how they can modulate the progression of the disease. And then the important and critical factor is really to be able to understand the disease itself at an organism, but to make this even more complex and to take this further, there is an additional group that is the multiscale factor group that aims to elucidate how other factors that are non-biological related, such as the socioeconomic status, uses, for example, of public transport, could directly impact on on COVID-19 progression, on the severity of the disease progression. And so this is a different aspect that is being integrated here in this SHAO, in this SHAO effort to bring several pieces together to be able to understand COVID-19. So in conclusion, as I said several times, we are providing understanding on SARS-CoV-2 infection and on COVID-19. And we are aiming at linking different AOPs and performing networks to address the different levels of complexity of the disease. And of course, that this is a huge endeavor, as you could see, and we are facing many difficulties to be able to integrate all of these in concise information, but also to be able to integrate, for example, the timeline of the disease, including long COVID. It's something that we are aiming for. The different layers of complexity that I referred to, the modulating factors, the multi-scale factors that could affect the disease. We are facing difficulties in integrating the different outcomes, whether it is a mild infection or a very severe outcome, and also trying to look at SARS-CoV-2 variants and how they change, affect, or alter what we are finding for the progression of the disease. And so this is what I wanted to share with you, but basically also tell you that we are looking for people to integrate and to assist in this SHAO event and effort, and we would welcome you to join one of our groups as well. So, and I welcome your questions. All the acknowledgements go, of course, to the crowd. Thank you very much.
Thank you, Maria João. Just to echo, we have a lot of people on the crowd here, or some people from the crowd in the audience here. So thanks for joining. Thanks for all the work that you've been doing in the Lung Hub group. It's been quite extensive to see over the past year how much activity, and it's fun to see how much activity there is and how much interest and truly engagement across toxicology, epidemiology, virology. It's very, very neat. Please enter your questions in the Q&A. Julia Filipskova is giving us an update in the Q&A that the text review is further along than last time. Maybe I could ask you, I know that I don't think at least you had a lot of experience with AOPs before coming in to CHOW, because it's really more of a sort of toxicology concept. And I'm curious, as a biomedical researcher, how you have, what are the kind of experiences that you've had applying the AOP concept to your field?
So it's actually a very good question, Christy. Indeed, I did not have a single experience about AOPs, the key events relationship, the molecular initiating events, all of those were unknown to me. And I thought that was actually a very neat strategy to look into every problem that we have at a molecular basis or a cellular basis or even an organismal basis in the lab. So in one of the publications, my publications of this year, I even attempted to have a figure that is totally inspired on AOP and on the AOP framework to highlight the important pathways and what are exactly the key events that are important. And in this case, I was looking at the research that we do in the lab, but to be able to try to pinpoint where the problem that we were investigating could make a difference or not. I suspect that it's very good from my side. It's really good to be able to look and understand the perspective of using AOP in other completely different frameworks from toxicology. And I think this actually has a lot of potential to open to many other fields. So I mean, using different viruses is something that I can immediately foresee, because they could be analogs of stressors we could think, but it's a very interesting concept.
That's great to hear that you started applying it in other work. I've seen a little bit here and there applying AOPs to things like, cardiovascular disease and other types of medical research. So it's interesting to see the concept kind of start to span out into other areas. Well, we don't have any more questions, so I think we will go ahead and close the webinar. We have thanks from the participants or from the attendees for your presentation. I would like to thank you for your presentation on behalf of ESTIV and ASCCT for coming and taking the time to talk with us about this project. Thanks as well to Marco. Thank you, Maria João, for spending some time during the summer. And we hope you will all join us for the next webinar. This will be recorded, so we'll be able to share the link once it's completed, once it's ready. And with that, I will say thanks a lot, and I'll have a great summer.